Darlings, as much as I wish that I can be there with you today, I can't. So, in best to be like physically present with you, I guess this is the best that I can do lah. At least got my face here, right? Okay. So yep, as in like let's just get straight into it. Yesterday, right, in the exploration of homeostasis, at least that there's some, knowledge and words in your mind. So this video is actually planning to concretize what we have learned yesterday, okay? So you have your notes and the slides as well in Google Classroom, but most importantly, I need the notes. So hopefully this video you can, put it one side while you pen down your notes together with me, okay? Can Okay, let's go. So as mentioned yesterday, right, as you can see here, right, the main three parts of homeostasis as mentioned yesterday is number one, okay, homeostasis itself as a principle. Why do we need homeostasis? What is homeostasis, right? Two, okay, the structure of your skin, the largest organ of your body, which you have already learned. And three, okay, temperature regulation, which is the crux of this chapter actually. So for today, we're just going to do 12.0. Oh, I need to flip the mirror. Okay, 12.0 and 12.1. Okay, the need for homeostasis and skin structure. So without further ado, let's go. The need for homeostasis. Okay, so as you already know, okay, as written below in the notes, what is homeostasis? Homeostasis is the maintenance of a constant internal environment. Okay, we've discussed yesterday that this is Cher's favorite definition in the whole book, or all of the definitions because it's short, sharp, and sweet. Okay, maintenance of a constant internal environment. Okay, so by now you should already know this already. Okay, now the thing is that the internal environment, right? Okay, so when you say maintain constant internal environment, what's that? Okay, internal environment refers to the conditions within the body of the organism, in this case us. So what is it in us that we need to maintain constant? Your blood and your tissue fluids, correct? So homeostasis allows us to be independent from changes in the external environment. Yesterday we talked about ectotherms and endotherms, right? Your cold-blooded and warm-blooded animals. So we are warm-blooded, right? So homeostasis for us allows us to be independent from whatever that's happening in the surroundings. So if suddenly, like if you go to a very cold country or even hotter country, we are still able to maintain a 36.7 degrees Celsius to 37 degrees Celsius within us, unlike the cold-blooded animals, where their body temperature will be the same as whatever the environment is. Okay, can So yeah, let's carry on. Okay, so why is homeostasis important, right? Okay, so number one, I need to point. Okay, yeah, number one, okay, it helps to keep your body temperature constant. Okay, remember that when we mention temperature, the first thing that should pump is enzymes. Okay, temperature and enzymes. Okay, and number two, over here, here, ensure that the composition of your body fluids, in this case like your tissue fluid, is kept within narrow limits. That means the pH as well as the water potential is maintained, which we also spoke about it yesterday already. Okay, so let's go with the first one. Importance of homeostasis. Why must we keep constant? Okay, so number one, okay, as mentioned here, maintain a constant temperature, because if there's changes in temperature, right, there's going to be enzyme inactivation. So inactivation refers to here, right, where the temperature is low, right, where if it's going to be low, the enzymes are going to be inactive. Or denaturation. You know that if it goes way beyond the optimum temperature, enzymes will start to denature. Okay, and that's not good because it's going to affect our body processes. Okay, so key point, we must maintain our body temperature. Now, number two, maintain your constant pH and water potential of the tissue fluid. Okay, the diagram above here, okay, should serve you as a reminder of what tissue fluid is, right? There's your capillary, right? And then the tissue fluid that's surrounding the cells, correct? Where the exchange of substances take place via diffusion. So if there's number one, okay, if there's going to be a sudden change in pH, there's going to be enzymes denaturation. So then just question is, right, then the tissue fluid got enzymes meh? Have or don't have? If you answer have, then I'm questioning, But by right, because the enzymes are found inside the cells, right inside, wait, I need to check myself, here, right, inside inside the cells. Am I right? So the thing is that when the cells are there, it's bathed in the tissue fluid, correct? That fluid with changes pH will affect the enzymes inside. And also dangerous because you want your cells to keep going, right? So you must make sure that there's no extreme changes in pH. Okay, so there's enzymes won't denature. Okay. Number two, we also spoke about water potential, right? If let's say if the water potential of the tissue fluid suddenly become very high, right? The body cells may burst because water molecules will start entering into the cells, and then as you know it will burst. Or if let's say suddenly, right, there's very low water potential, then the cells may crenate, which also will affect the body function. Okay, so long story short, oh wait, sorry, before I come here, is that we need homeostasis for two reasons, right? Maintain constant temperature as well as maintain pH and water potential of your tissue fluid. Okay? Okay, now here, as mentioned yesterday also, homeostasis and negative feedback must always appear with one another. Remember that bio is the art of accuracy and inquiry, right? So you must be accurate and ask a lot of questions. So homeostasis and negative feedback. Now, yesterday was the first time that we came across this term, okay? So the thing is that today here, okay, let's be very clear and familiar of like, what's this whole thing, like positive, negative feedback, what? Okay, let's just talk about it here. Okay, so let's carry on. Okay, before we carry on talking about negative feedback, let's be clear of some new words that we need to learn first. Before we jump in. Okay, so number one, stimulus. Okay, can you like whisper or like say it to yourself? Stimulus. Okay, stimulus is any change in the internal environment for this case, okay, from your normal condition. Right? For example, suddenly, blood glucose level change, right? We eat a meal. Or suddenly, the water potential of the blood may change because we drink a lot of water. So all these are stimulus. Okay? So you have just learned now why it's important to maintain a constant internal environment, right? You know, your enzymes will start to denature or become inactive, or it can affect the water potential of the cells, right? They may burst or crenate. So, in homeostatic control, your body reacts to bringing an opposite effect to any change detected so that the system returns back to normal. I.e., let's say if, your blood sugar level or glucose level is normal, if let's say suddenly, increase, then homeostatic control have to bring it back to the normal, right? Back to normal. Or let's say if it decrease, you need to increase it back to normal. This is called homeostatic control. Okay, body reacts an opposite effect to any change detected. Okay, so your next key definition. Okay, so negative feedback process. Okay, so we know stimulus already. Okay, negative feedback process. It is the process by which your body counteracts change in the internal environment and restores it back towards the set norm. Need me to say again? I don't know if there are too many new words there though. So the negative feedback process is the process by which your body counteracts change in the internal environment and restores it back to normal. So if any deviation or any change, okay, the negative feedback process will have to change it back to the normal. Okay, so sense organs or receptors will help to detect this change in the body condition. Okay, so let's give an example. If not, I'm talking very many big words here. So as you can see, after a big meal, right, we ate, what happens? Okay, your blood glucose level will increase. Am I right? Suddenly, it increases all the way up, very, very high. So that is a stimulus. Okay, it's a term that you must write in your essay if question comes out. Stimulus. Okay, the blood glucose level goes high. So then what happens? Okay, then now this one, the receptors. Okay, receptors are sense organs. In this case, the pancreas, right, will detect this change. Hello, right? So when it detects this change, a corrective mechanism will take place, which you should know by now, right? That one later we'll cover. Okay, so the pancreas will actually detect this change and then it will secrete insulin. Okay, not glucagon, insulin, right? Then blah, 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 blah. Then after that, it will bring it back to become normal, right? So it's one complete loop of the negative feedback process. Okay, so this is the important one here. Please start this diagram. Okay, so this is actually the principles of the negative feedback control loop or the negative feedback process, right? So first off, there's always a normal, right? After that, there's stimulus. It can be increased or it can be decreased. Okay, increase or decrease. Okay, so there must be a stimulus. Okay, after that, what happens? A receptor must detect the stimulus. Okay, these are usually sense organs, could be your pancreas, or your brain or something. Okay, your receptor will have to detect the stimulus. Then there's a self-regulatory corrective mechanism. You can see here, that means things will happen in your body lah, okay, to change things back to the normal. So in this case, if it decrease, okay, then now the condition will increase. Then if the condition increase, then the condition will decrease. Basically, bring it back to the normal. Okay, so stimulus, receptor, self-regulatory corrective mechanism. That means like the processes that happen to bring it back to normal. And last but not least, negative feedback. Number 4, negative feedback. Okay, can So let's just go on first. Okay, if the end don't understand, then we ask questions. Okay, so first, norm, right? Normal condition or set point. Great. If things are kept here, awesome. Okay, then there's stimulus, right, which is change in the internal environment, right? I only put the example for increase here, but it could be decrease as well. So condition increase or decrease beyond the norm, there's a change in environment and a stimulus occurs. Okay? All right. Then we have receptors, right? Your sense organs that will detect this stimulus and send signal to the control center. Okay? So stimulus, receptor. Then your self-regulatory corrective mechanism brings about a reverse effect to the stimulus. So if something increase, this corrective mechanism will decrease it back to normal. If it decrease, these mechanisms will increase it back to normal. Okay, brings about a reverse effect to the stimulus. And last but not least, negative feedback to the receptor. This is the one. Okay, it causes the corrective mechanism to stop once the set point is reached. Okay, so 4 things. Okay, everything normal, great. Okay, then the keywords are stimulus, something changes. Receptor will detect the change. The self-regulatory corrective mechanisms to actually change back things to normal. And finally, negative feedback. So you all keep asking us, what's negative feedback? So negative feedback is actually when the condition increase or decrease back to normal already, right? It causes the corrective mechanism to stop. Because if it doesn't stop, like for example, in this case, then the condition will keep decreasing and decreasing and decreasing, and then it will go way below normal. But no, that's not what you want. It's just you want decrease back to normal and then, okay, stop and maintain there. So the negative feedback is when it starts to decrease, then straight away it tells the receptor, hey, enough. Then, boom, then it will stop there at the normal. Okay? So, do we need to remember all these like long definitions? No need. The main key point here, okay, is the keywords norm, stimulus, receptor, the self-regulatory corrective mechanism, okay, and negative feedback. Okay, so the 5 keywords you must know, and then we apply it to a context. Okay, Okay, before we go on to the examples, if you need to pause or rewind, you go ahead. Okay, but as in like the main thing is that, so once you know the keywords, right, then you need to apply to the 3 examples, which I'll be coming on in just a second. Okay, so okay, I talk about the common misconception first. So yesterday we spoke about positive feedback also, right? There's negative feedback, there will be positive feedback. Now, positive feedback is not really in your syllabus, like I mentioned, but since we came into that, right? Okay, let me just explain. Okay, now, negative feedbacks, you should by now know, right, is to basically keep resetting things back to normal. Correct or not? Whether it increase or decrease, it must bring it back to normal. But for positive feedback, it amplifies the change. So let's say if something it increases, right? You keep increasing and keep increasing. You never bring it back to normal. You get what I mean? Just keep increasing or keep decreasing. So an example, as you've seen yesterday, correct, could be when a woman is giving birth, and then like there's contractions in the muscles to squeeze the baby out in the cervix, right? Then more hormones to stimulate the contraction. That's an example of positive feedback, right? Or another example that's stated in your textbook is that the positive feedback occurs during lactation. That is secretion of milk from the mammary glands to feed the baby. So down there also positive feedback. Hey, more milk for the baby. Like the hormones will cause the cells there to more milk. So it keeps going. So that's positive feedback. Okay, Yonje, I think you were confused with this yesterday. So do not confuse stimulus with positive feedback. So for example, right, okay, this is the normal. When suddenly a stimulus causes increase, right? Maybe like blood sugar level increase beyond the normal. This is still part of negative feedback. This is not positive feedback because we never keep increasing. We are trying to bring things back to the norm. Okay, so do not confuse an increased stimulus with positive feedback. Okay? If still confused, please ask me after school, like text me or something. Yeah, if not, as in I'll ask you guys next week on this. Okay? So Everyone okay so far? Okay, hope you guys are alright. Okay, for homeostasis, as I said, so now it's applying this like stimulus, receptor, blah 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 into the examples. This is the more important part. Okay, so you need to know three. Number one, the first two, you have already learned it before. Okay, number one, how to regulate blood glucose concentration, right? To maintain it at a good level. That's your first example that you must know. You learned it before already, actually. Number two, how to regulate your blood water potential. You've learned this in excretion. Okay, so maybe holiday, come back, forget a bit. Now's the time to revise. And number three, regulating temperature. This one we do it next week. Okay, the crux of the chapter of homeostasis, we do it next week. But these two above, we need to know. Okay, alrighty. So let's go for the first one, regulating blood glucose concentration. Okay, now let's start. Okay, so the story goes, because bio is all about stories, right? Number one, there's normal blood glucose level. Everything normal, right now. Okay, so you see, you can either increase, like after you eat a heavy meal, right? You increase, or you can decrease, right? Oh, there's an error in the notes. Okay, wait, there's an error in Mr. Cho's notes, but as in, can you all change it? I can't change it on the Zoom. Decrease in blood glucose concentration is like after exercise or after fasting. Okay, please change it. Okay, never mind. I'll change it after the thing. But yeah, okay. So there can be an increase in blood glucose concentration, or there can be a decrease in blood glucose concentration. So this one should be fasting, okay, not after a heavy meal. Then, so what happens when the blood glucose levels change? The islets of Langerhans, okay, as you can see in the picture here, okay, I put a picture already, okay, so the pancreas, right, inside, there's a special group of cells, okay. Ignore all these like, very, very cheap things. Just this area where Mr. Cho is circling, right, is the islets of Langerhans. A group of cells, okay, in the pancreas are stimulated. These cells produce insulin and glucagon. Okay? The cells in the islets of Langerhans, write it down, okay, produces insulin or glucagon. Got it? Okay, so this one is if the blood sugar level increase beyond the normal, right? We say this one first. So if let's say if it increases, then what's the self-regulatory corrective mechanism? Okay, the islets of Langerhans will secrete insulin into the bloodstream, right? So then insulin will travel by blood and causes the there's a lot of error here. Can you please delete this? And it causes the liver, okay, it causes the liver to convert excess glucose into glycogen. I'll change it in the slides later, okay, but it's wrong here. Causes the liver, okay, to convert excess glucose into glycogen. Glycogen is then stored in the liver and muscle cells. Okay, it also increases the thing is blocking, increases the permeability of the cell surface membrane, okay, to take in glucose. So all these are corrective mechanisms. Okay? Meanwhile, on the other hand, if let's say there's if there's a lack of glucose in the bloodstream, then it will secrete glucagon into the bloodstream, right? Then it will cause the liver and muscles to convert the stored glycogen into glucose. Okay, say one more time. Over here, this one, okay, is actually liver, not islets of Langerhans. I'm sorry for the error here. Okay, carrying on. So what happens? As you can see from the respective one will cause the blood glucose level to decrease, one will cause it to increase back to the set norm. Then include in this line, right? So after negative feedback, then after when the blood glucose concentration decrease or increase, this will then provide a feedback to the receptor to reduce insulin production or to reduce glucagon production respectively. That means, hey, stop, don't want any more insulin or glucagon now. This is the step that is crucial to put in your answer. And then the blood glucose level will return back to normal. Okay? If you need to replay it. If not, then we are good. By right, this should be a refresher. And I'm sorry for the error though. I think some copy and paste error. I'll make sure you guys get the corrected ones in the slides. Alright, okay. So with that, we've completed one example already. So this is your second example, regulating blood water potential. Okay, this one is in excretion. Now, one thing here, okay, take note of this, okay, that the terms blood water potential and there's also osmotic pressure. Okay, you all cannot be confused. Okay, so this is something new. So pay attention. Now, blood water potential, right? Okay, is inverse from osmotic pressure. So that means, okay, let's say in your blood there's more water, right? So if there's more water, the blood water potential will increase. But with that being said, okay, you can either say that or the osmotic pressure will decrease. You get what I mean? So if you say blood water potential increase, means you'll learn this new term, osmotic pressure will decrease. But if the blood water potential decrease, osmotic pressure will increase. Personally, Mr. Cho prefer to explain this using the terms blood water potential than osmotic pressure. But I'm putting it both here because as in like the rest of the bio teachers are teaching that too. But yeah, because it's about what you understand. So it's your preference as well. Okay? Okay, come. Let's go. So same thing. We start with normal blood water potential first. If you can draw arrows down like between each box can, because I can't put it in the text box. Okay, so it could be the case where there's a decrease in blood water potential, right? For example, like you sweat. Wow, you go for exercise, you sweat, correct or not? So there's a decrease in blood water potential, or you drink a lot of water on the other hand, right? There's a large intake of water, right? So in that you increase the blood water potential, correct? So I don't use osmotic pressure here to explain, okay? But for convenience sake, it's actually the reverse. So if let's say there's a decrease in blood water potential, means increase in blood osmotic pressure and vice versa. Okay, so I'm just going to use blood water potential to explain. Okay, so what happens when the blood water potential increase or decrease? Then the osmoreceptors in the hypothalamus in your brain will detect the change. So these are the dudes that's in charge of actually producing your ADH, your antidiuretic hormone. Okay, so again, let's take it left first, then we take it right? So if let's say there's going to be, let's say there's going to be a decrease in blood water potential because you sweat a lot, then how? Okay, so the corrective mechanism is there's going to be more ADH to reabsorb back the water, right, at the collecting ducts in your nephrons, right? So to increase back the blood water potential back to normal. Meanwhile, on the other hand, correct, let's say you drink a lot of water, then how? Then need to secrete less ADH, right? So, by the pituitary gland, and then will cause less water to be reabsorbed back in the collecting duct. So more water goes out in the urine. So your blood water potential will decrease, okay, as in the blanks there will decrease, right? So, okay, meanwhile, one increase, what the other one decrease, what's going to happen? Now, the important line again, the negative feedback line, right? So negative feedback will occur. So as water potential of the blood will increase, feedback, okay, is sent to the receptor, then to, in this case, will be reduced. In the other case, will be increased, okay, the ADH production or the secretion, okay, bringing things back to normal. Okay, so my last thing before you go for your quick check. So can you see the main thing is like you must get is the keywords like norm, stimulus, receptor, corrective mechanism, and negative feedback, and then what are the two examples and how it relates. Okay, so in this case, for example, the norm is water potential. So the stimulus is a change in the water potential, right? Then the receptors are the osmoreceptors. Osmo means detect water in the brain. That's your receptors. And then things happen in the body. The processes that happen in the body are your mechanisms. And finally, negative feedback to when things go back to normal already, tell you, hey, stop. Okay, in this case, will be the feedback will be sent to the osmoreceptors in the brain. Okay, so give a quick check 12.0 a try. Okay, give it a try, and then give 12.0 a try, and then after that, I won't be going through the answers here. Okay, next week, either during HBL or the week 3 that I see you, then I'll go through this with you. Please do, okay, because when you write, you'll be able to concretize your knowledge. Okay, and then so two last naggings here. Number 1, so if you take a look at your quick check 12.0 question 2, right, make sure that when you explain, I asked about blood water potential, right, the keywords should be there. So like for example, like stimulus, something, then receptors, so the osmoreceptors, something, corrective mechanism, then negative feedback. So all these words must appear in your answer. Okay, okay, and after that, okay, so we will have structure of the human skin. Okay, for that one, right, I think it is pretty self-explanatory on the slides. Okay, so use the slides to copy into your notes first and read through and understand. Okay, we'll take it when we see each other next week for HBL. Okay, I really wish I was in class with you guys, but as in can't wait to see you all soon already. So finish up your quick check 12.0. Okay, 12.1, go and read and finish up 12.1 as well. So next week when we start, we straight away can go on thermal regulation during HBL already. Okay, I'll see you guys soon. Hope this video helps. Okay, bye.